Alright class, now we are starting a new lecture, and the focus is shifting from cell wall and membrane to something equally important, protein synthesis. Specifically, we will study drugs that target the 50S ribosomal subunit. Remember, ribosomes are essential for bacterial survival because they are responsible for making proteins. If protein synthesis stops, the bacteria cannot grow or survive. In this lecture, we will cover 4 major groups: macrolides, lincosamides, oxazolidinones, and amphenicols. Each of these drugs binds to the 50S subunit, but at slightly different sites and steps, so their mechanisms and clinical uses differ. Some block peptide elongation, some prevent initiation of protein synthesis, and others inhibit peptidyl transferase activity. So keep one concept in mind as we move forward: different drugs, same target, 50S, but different mechanisms and clinical roles. By the end of this lecture you should be able to understand where different drugs bind on the 50S ribosome and how they block protein synthesis. Explain key mechanisms like translocation inhibition, initiation block, and peptidyl transferase inhibition. You should also be able to connect this knowledge with resistance mechanisms and apply it in clinical decision-making, especially for respiratory and soft tissue infections. Alright class, now let's build a clear story in your mind. Bacteria survive by making proteins, and proteins are made on the ribosome, which has 2 parts, 30S and 50S subunits. Today we are focusing on the 50S subunit, which mainly controls peptide bond formation and elongation of the protein chain. So if we block this part, the bacteria cannot make functional proteins, and without proteins, life stops. Now different drug classes target different steps on the same 50S subunit, and that is the key to understanding this table. Let's go step by step in the order of protein synthesis. First, think about elongation and movement of the ribosome. This is where macrolides like azithromycin act. They block translocation, meaning the ribosome cannot move forward on mRNA. So the protein chain cannot grow further. Clinically, these are used in respiratory infections, CAP, and cover gram-positive and atypical organisms. Next, we move to peptide bond formation. Here come lincosamides, like clindamycin. They inhibit the formation of peptide bonds, so amino acids cannot be linked together. These are especially useful in anaerobic infections and some gram-positive infections. Now go even earlier, the initiation step of protein synthesis. This is where oxazolidinones, like linezolid, act. They prevent the formation of the initiation complex, so protein synthesis cannot even start. That is why they are very powerful against resistant organisms like MRSA and VRE. Then we have streptogramins, which also act on the 50S but in a slightly different way. They interfere with translation and are particularly useful in VRE infections. Think of them as disrupting the whole process rather than a single step. Now another important class is chloramphenicol, which inhibits the peptidyl transferase enzyme, the enzyme directly responsible for forming peptide bonds. This is a broad-spectrum drug but used selectively due to toxicity, for example in typhoid fever. Then comes fusidic acid, which is slightly different. It does not directly bind the ribosome but inhibits EFG, a factor required for translocation. So indirectly, it still blocks protein synthesis. It is mainly used in staphylococcal infections, especially bone infections. Finally, chloromutilins like lefamulin act again on the peptidyl transferase center, PTC, but at a unique site. These are newer drugs and used mainly in community-acquired pneumonia. So if you summarize this whole table in one line, all these drugs target the 50S ribosome, but each blocks a different step: initiation, elongation, peptide bond formation, or translocation, and that determines their clinical use. Alright class, now we start one of the most commonly used and clinically important groups, macrolides. These are called macrolides because they have a large lactone ring structure, which is important for their function. The classical drugs include erythromycin, clarithromycin, and azithromycin. Erythromycin is the prototype but has more side effects, especially GI upset due to model and receptor stimulation. Clarithromycin and azithromycin are improved versions, more stable, better tissue penetration, and better patient compliance. Now look at the advanced modifications. Azithromycin belongs to azaleads, where a nitrogen is inserted into the ring, giving it a long half-life and excellent tissue distribution. That is why it is widely used in respiratory infections. Then we have ketolides like telithromycin, which bind more strongly to the ribosome and can overcome some resistance mechanisms, especially mediated resistance. Now coming to mechanism, which is very important. Macrolides bind to the 23S rRNA of the 50S ribosomal subunit. They block translocation, meaning the ribosome cannot move from a site to P site. Also, they block the exit tunnel, so the growing peptide chain cannot come out. Because of this, protein synthesis stops. These drugs are generally bacteriostatic, but can be bactericidal at higher concentrations. Clinically, these drugs are very useful in respiratory infections, especially for atypical organisms like Mycoplasma, Chlamydia, and Legionella. They are also used in H. pylori therapy and as alternatives in penicillin-allergic patients. But you must remember adverse effects, QT prolongation, GI upset, and important drug interactions due to 450 cypriot pounds inhibition, especially erythromycin and clarithromycin. So the key idea is, macrolides block translocation on 50S right-pointing arrow stop protein synthesis right-pointing arrow mainly used in respiratory and atypical infections. Now we move to lincosamides, and the most important drug here is clindamycin. This is a very commonly used drug, especially in hospital settings. First, understand the mechanism. Clindamycin binds to the 50S ribosomal subunit, specifically at the peptidyl transferase center. Because of this, it blocks peptide bond formation. So the bacteria cannot join amino acids to form proteins. In simple words, the protein chain cannot grow. This stops bacterial growth, so the drug is mainly bacteriostatic. Now coming to clinical use, this drug is very important for anaerobic infections. It works well against organisms like Bacteroides. That is why it is used in intra-abdominal infections, dental infections, and abscesses. It is also a good option for skin and soft tissue infections, especially in patients who are allergic to penicillin. Another important point, clindamycin can suppress toxin production, so it is used in severe infections like necrotizing fasciitis and toxic shock syndrome caused by group A streptococcus. Now very important, adverse effects. Clindamycin is strongly associated with Clostridioides difficile infection, CDI. Why? Because it kills normal gut flora, especially anaerobes, and allows C. difficile to overgrow. So clinically, whenever you see diarrhea after antibiotic use, always think of this possibility. Now let us discuss resistance, which is very important for exams and clinical practice. Resistance can be due to gene-mediated methylation of the ribosomal binding site. This can be constitutive or inducible. This brings us to the D-test. This is used in the lab to detect inducible resistance. If a bacterium appears sensitive to clindamycin but resistant to erythromycin, we place both antibiotic discs near each other. If the clindamycin zone becomes flattened, D-shaped, near erythromycin, it means inducible resistance is present. In that case, do not use clindamycin, because treatment can fail. So the key idea is, clindamycin blocks peptide bond formation on 50S right-pointing arrow good for anaerobes and skin infections right-pointing arrow but high risk of C. difficile and inducible resistance must be checked, D-test. Alright class, now we move to a very interesting group, streptogramins. These drugs are unique because they work in combination, and their strength comes from this synergy. First, understand the concept of synergistic combination. Streptogramins are usually given as a combination of quinupristin, 30%, and dalfapristin, 70%. Individually, they are only bacteriostatic, meaning they just stop bacterial growth. But when combined, they become bactericidal, meaning they can actually kill bacteria. This is very important clinically. Now let us understand the mechanism step by step. These drugs bind to the 50S ribosomal subunit, but in a sequential manner. First, dalfapristin binds, and this changes the shape of the ribosome. This conformational change increases the binding of quinupristin. Then quinupristin binds more strongly and together they block peptide elongation and release of the protein chain. So protein synthesis is effectively stopped at multiple steps. This dual action is the reason for their strong effect. Now coming to the spectrum, these drugs are mainly active against gram-positive bacteria, especially resistant ones like MRSA and VRE, Enterococcus faecium. However, an important limitation is that they are not effective against Enterococcus faecalis, mainly due to intrinsic resistance mechanisms like efflux pumps. So always remember, VRE, faecium, yes, faecalis no. Finally, we discuss adverse effects, which limit their use. Patients often experience muscle pain, myalgia, and joint pain, arthralgia. There can also be venous irritation, especially with IV use. Another important point is that they inhibit CYP3A4, so they can cause significant drug interactions. So the key idea is, streptogramins work as a synergistic pair on 50S right-pointing arrow sequential binding blocks protein synthesis strongly right-pointing arrow mainly used for resistant gram-positive infections like MRSA and VRE, but limited by side effects. Now we come to a very important and slightly different group, oxazolidinones, mainly linezolid and tedazolid. These drugs are special because their mechanism is different from all other protein synthesis inhibitors. First, understand the mechanism. These drugs bind to the 23S rRNA of the 50S ribosomal subunit, but instead of blocking elongation like macrolides or clindamycin, they act very early. They prevent the formation of the 70S initiation complex. This means the ribosome cannot even start making protein. So in simple words, protein synthesis is stopped at the starting step. Because of this unique action, they're very useful against resistant bacteria. Now coming to clinical use, these drugs are mainly used for gram-positive resistant organisms. They are very effective against MRSA and VRE, both E. faecium and E. faecalis. Another important advantage is their excellent lung penetration, so they are widely used in pneumonia, including hospital-acquired infections. Also remember, linezolid has 100% oral bioavailability, so we can easily switch from IV to oral without losing efficacy. Now let us discuss adverse effects, which are very important clinically. The most common is myelosuppression, especially thrombocytopenia, so patients need regular CBC monitoring, particularly if treatment is longer than 2 weeks. Another serious effect is neuropathy, including optic neuropathy, especially with prolonged use. A very important point, linezolid is a weak MAO inhibitor. Because of this, it can interact with drugs like SSRIs and SNRIs, leading to serotonin syndrome. So always be careful with drug history. Finally, a small comparison, tedazolid is a newer drug. It has once-daily dosing and tends to have less myelosuppression and fewer drug interactions compared to linezolid. So the key idea is, oxazolidinones block initiation of protein synthesis right-pointing arrow very effective against MRSA and VRE right-pointing arrow but require monitoring for bone marrow suppression and drug interactions. Now we discuss chloramphenicol, a very powerful but clinically restricted drug. It is important conceptually and also for exams. First, understand the mechanism. Chloramphenicol binds to the 50S ribosomal subunit and specifically inhibits the enzyme peptidyl transferase. This enzyme is responsible for forming peptide bonds between amino acids. So when this enzyme is blocked, the protein chain cannot grow. In simple words, amino acids cannot join right-pointing arrow protein synthesis stops. Because of this, chloramphenicol is mainly bacteriostatic, although it can be bactericidal against some organisms. Now coming to the spectrum, this drug is broad spectrum. It works against gram-positive, gram-negative, anaerobes, and intracellular organisms. That is why historically it was widely used. It is particularly effective against organisms like H. influenzae, Neisseria meningitidis, and Streptococcus pneumoniae. However, despite this wide coverage, its use is very limited today. The reason is toxicity, which is very serious. The most important toxicity is aplastic anemia. This is rare but irreversible and potentially fatal. It is not dose-dependent and can occur unpredictably. That is why this drug is avoided unless absolutely necessary. Another important condition is gray baby syndrome. This occurs in neonates because their liver enzymes are not fully developed, so they cannot metabolize the drug properly. This leads to accumulation, causing hypotension, cyanosis, and gray discoloration of the skin. There is also dose-dependent bone marrow suppression, which is reversible. This can cause anemia, leukopenia, or thrombocytopenia. Because of these toxicities, chloramphenicol is now used only in specific situations. For example, in severe infections where no safer alternatives are available, such as certain rickettsial infections, or meningitis in patients with severe penicillin allergy. So the key idea is, chloramphenicol blocks peptidyl transferase on 50S right-pointing arrow, very broad-spectrum right-pointing arrow, but severe toxicity, aplastic anemia, gray baby syndrome, limits its use to special cases only. Alright class, now we discuss how bacteria become resistant to drugs acting on the 50S ribosomal subunit. All these antibiotics share the same target, so bacteria mainly use a few common strategies to escape their action. The most important mechanism is target modification. Bacteria produce an enzyme encoded by the gene. This enzyme methylates the 23S rRNA of the 50S subunit. Because of this modification, antibiotics can no longer bind properly. As a result, the bacteria become resistant. This leads to what we call MLSB resistance, which includes macrolides, lincosamides, and streptogramin B. This type of resistance is usually strong and clinically significant. Now, sometimes this resistance is not always active. It can be inducible. In such cases, the bacteria may appear sensitive to clindamycin in routine testing. However, when exposed to macrolides, the resistance mechanism is activated. To detect this, we perform the D-test. If the zone of clindamycin becomes flattened near the erythromycin disc, forming a D shape, it means inducible resistance is present. In this situation, clindamycin should not be used, even if the report shows sensitivity. Another mechanism is active efflux. This is mediated by the MSR gene, which produces pumps that remove the antibiotic from the bacterial cell. Because the drug is pushed out, it cannot reach effective levels inside the cell. This mainly affects macrolides and streptogramin B. An important point is that clindamycin is not affected by this mechanism, so it can still be used in such cases. Finally, there is a more advanced mechanism involving the CFR gene. This also causes methylation of the ribosomal target but at a different position. This leads to a broader resistance pattern known as pH-LOPSA resistance, affecting multiple drug classes including chloramphenicol, lincosamides, oxazolidinones, pleuromutilins, and streptogramin A. This is clinically important because it can cause resistance to drugs like linezolid, which are often used as last-line therapy. This gene is often carried on plasmids, so it can spread easily between bacteria. So the key idea is simple. Bacteria resist 50S inhibitors by modifying the ribosome, pumping the drug out, or activating resistance when exposed to antibiotics. Clinical interpretation, especially D-test, is very important for correct treatment decisions. Alright class, now we connect everything we have learned with real clinical cases. This is where mechanism, pharmacokinetics, and patient condition all come together. First case is MRSA pneumonia. A 65-year-old patient has hospital-acquired pneumonia, and culture shows MRSA. In such cases, the preferred drug is linezolid. The reason is very important. Linezolid has excellent lung penetration, especially into the epithelial lining fluid. This means the drug reaches very high concentrations at the site of infection. In comparison, vancomycin has relatively poor penetration into lung tissue. So even though both cover MRSA, linezolid is often preferred in pneumonia. This is a classic example where pharmacokinetics guides therapy. Now the second case is streptococcal toxic shock syndrome, TSS, caused by group A streptococcus. Here, the problem is not only bacterial growth but also toxin production. The treatment includes penicillin plus clindamycin. Penicillin kills actively dividing bacteria, but in severe infections with high bacterial load, some bacteria are in stationary phase and less sensitive. This is called the eagle effect. Clindamycin is added because it blocks protein synthesis, and therefore stops toxin production. This is very important in TSS because toxins are responsible for severe symptoms and shock. So even if bacteria are not rapidly dividing, clindamycin still works by reducing toxin production. So the key idea is, in MRSA pneumonia right-pointing arrow choose linezolid due to better lung penetration. In toxin-mediated infections like TSS right-pointing arrow add clindamycin to suppress toxin production. Alright class, now let us summarize what we have learned about 50S ribosomal inhibitors in a simple and connected way. First, focus on target specificity. All these drugs act on the 50S ribosomal subunit, but they block different steps of protein synthesis. Macrolides and streptogramins mainly block translocation, oxazolidinones block initiation, and drugs like chloramphenicol and clindamycin inhibit peptidyl transferase. So even though the target is same, the exact step is different. Now coming to resistance genetics. Bacteria mainly resist these drugs by modifying the ribosome using ERM genes, pumping drugs out using MSR genes, or by broader resistance through the CFR gene. These mechanisms are very important because they directly affect treatment decisions. Next is clinical significance. These drugs are very important in treating resistant gram-positive infections, especially MRSA and VRE. Also remember, some drugs like clindamycin are useful not only for killing bacteria but also for reducing toxin production, which is critical in severe infections. Now think about adverse effects and monitoring. Each drug has its own important toxicity. Macrolides can cause QT prolongation, linezolid can cause bone marrow suppression and serotonin syndrome, and clindamycin is associated with C. difficile infection. So monitoring is essential during treatment. Finally, therapeutic application. Drug selection is not random. It depends on the site of infection and drug properties. For example, linezolid is preferred in pneumonia because of good lung penetration, while clindamycin is useful for anaerobic infections and toxin suppression. So the final idea is, same target, 50S, different mechanisms, important resistance patterns, and careful clinical selection based on patient and infection.